Hey everybody! For this video, we're going to go ahead and cover our temperature and pH testing that we're going to be doing in class. And considering that the techniques that we use to basically test for pH or temperature are very similar, again, we'll just kind of go ahead and cover it in one video rather than making two separate ones. So let's start out kind of on the temperature side of things and kind of talk about the 5 different temperature groups that we have for bacteria. Our first group is what we call the psychrophiles. And psychrophiles are cold-loving organisms, generally living somewhere around -8 degrees Celsius to about 18 degrees Celsius. Again, they tend to like it very cold, so generally down in that freezing temperature. Our next group is what we call the psychotrophs. And psychotrophs are also cold-loving organisms, but they can live in a bit warmer environments. So they tend to be more around 0 degrees Celsius to about 30 degrees Celsius. And within this group here, an example of an organism that we could be concerned about could be something like Listeria monocytogenes, which is an organism that can live inside of refrigerators again, which is about 4 degrees Celsius. And Listeria monocytogenes, again, we're worried about in things like food poisoning. You can get listeriosis from it. And then we're also worried about it in pregnancy, where that you can basically get it from eating like lunch meats and things like that hasn't been thoroughly processed or hasn't been cooked, basically. But it can cause certain kinds of like complications, basically, with pregnancy. So that's where we're just an example of an organism that we're concerned about within this group. The next group that we have is then the mesophiles. And the mesophiles are kind of our body temperature organisms. They like to live between 10 degrees to about 48 degrees Celsius. And this is by far the group that we are the most concerned with, again, because they live in body temperature. So that's going to be the group that has the most organisms that are going to make us sick. Going up a little bit on the temperature scale, we can then have the thermophiles. The thermophiles are now heat-loving organisms. They like to live in environments from about 40 to 72 degrees Celsius. And then we have the hyperthermophiles. And our hyperthermophiles are basically going to be the ones that like extreme heat. So somewhere around 65 degrees all the way to above boiling or 110 degrees Celsius. So we generally don't interact with hyperthermophiles unless we're doing research and we're going to specifically go looking for them. But with the thermophiles, there might be some of those in certain types of like canning processes that we might see. And if they are allowed to live in some of these different environments, it can basically spoil food sometimes. But those are the basic temperature groups that we have and that we can test for in our lab setting. Moving kind of to the pH side of things, there are 3 basic pH groups that we can have. And depending on who you ask, the specific ranges that these fall under kind of varies. So we're going to go with what our lab book says, and that's going to be the numbers that we're going to use. So under the acidophile group are acid-loving organisms. They're going to be any bacterium that live or prefers an environment at pH 5.5 and below. Our next group are then going to be the neutrophiles, and the neutrophiles like a neutral environment, so anywhere between pH 5.5 and 8.5. And then we have our alkalophiles, and the alkalophiles are the ones that like a basic environment or an alkaline environment, and they're going to live anywhere between pH 8.5 and above, basically. That's where they're going to prefer their specific environment. Now, in terms of the way that we actually do this testing, if we want to figure out if an organism falls within any one of these specific groups, what you would end up doing is that, let's go ahead and I guess do the temperature part first. The materials you would need to do the temperature experiment, you're going to need a loop to do your transfer. You're going to need the Bunsen burner and the striker in order to do your aseptic technique. You are going to need your sample, and the sample can be either from a broth plate or a slant. It doesn't really matter, just any type of sample that you might want to test. And then normally you're going to need your different temperature environments. So in this case here, we tend to test 5 basic temperature environments. We normally do a 0 degrees, which would be our freezer. We do a 4 degrees, which would be inside of a refrigerator. We do a 25 degrees, which is just room temp, so somewhere out in the lab. We do a 37 degree, which is body temperature. And then we kind of hit up a 55 degree as well with a 55 degree incubator. So I guess I should have mentioned the 37 degree would be a 37 degree incubator. So in order to do your temperature testing, again, you will need those 5 environments. The last thing you're then going to need is going to be 5 broths in order to do your testing with. And the basic procedure for it, again, is that you're just going to do a standard inoculation of those 5 broths. And then from there, you're going to put them in the 5 temperature environments that you have, wait your 48 hours to a week or so, depending on our incubation period, and then take a look at them. And we'll kind of get into the scoring part here in a little bit once I've done talking about the pH section. So again, those are the materials that you would need for your temperature setup and kind of the basic procedure. Again, inoculate this stuff, let it incubate, and then look at it. In terms of the materials that you would need for your pH experiment, again, it's going to be very similar. You're going to need your loop to do the inoculation with. You're going to need your Bunsen burner and striker for your aseptic technique. You're going to need your sample again, doesn't matter where it's from, broth, plate, slant, doesn't really matter. And then you're going to need your different pH tubes. So in this case here, what you're going to get is you're going to get 5 different kinds of pH tubes. You're going to get one that's a pH 3, one that's a pH 5, one that's a pH 7, one that's a pH 9, and one that's a pH 11. And basically, depending on the number of samples that you're going to inoculate, you would then need a full set of those tubes per sample that you're going to inoculate. But let's just assume again that we're just testing one of them. You would get your 5 tubes in terms of the procedure, get the 5 tubes, inoculate them with your sample, and then look at them again after that 48 hours to a week incubation period. And when I keep saying again, we're going to be looking at them. When we're scoring our specific organisms based on which type of environment that they like the best, we can use a couple different mechanisms in order to basically score them. We can do a very basic scoring where we just look for growth. So in this case here, I would just take my tubes either from the temperature experiment or from my pH experiment, and I would just look at, is any one of them turbid? And if they're turbid, I can say they can grow in that environment. That's the very basic way that we can do those types of scoring. The next level up from that is basically doing a turbidity reading, and that is what these tubes are for right here, or I guess sort of a turbidity scoring, I should probably say, is a little bit more accurate. And what I have right here again are 6 tubes. They all have a score on them, so the score basically goes 0, 2, 4, 6, 8, and 10. And when we look at them, they all have different levels of cloudiness to them. So again, the 0 one is basically just an uninoculated tube, and then the other ones again just become more and more cloudy as we go up through the different levels. So what we can do again to make this a little bit more specific is that we can compare our tubes to the ones that we have incubated, and we can then give them a score either again at each of the different temperature levels, or we can give them a score again at each of the different pH levels. And in that way we can kind of see the more turbid they are, the more they grew in that environment, and the more likely it is that that is the optimum area where that they like to live. So again, it gives us a little bit more of just a better way basically of scoring it rather than just saying yes or no, they grew in those specific environments. So this is the one that we're going to be using in our class in order to score our tubes and basically try to figure out what is the optimum pH and optimum temperature for the specific organisms that we're going to be looking at. It will also allow us to do a bit of graphing and things like that depending on the specific data that we get. Now there is one more way to actually score it, and that is by using what we call a spectrophotometer. And a spectrophotometer, and it's this machine right here, and it's basically just a tool that basically shines light through your sample, and it can sort of determine based off of using a blank. You shine the light through the blank, and that basically blocks a certain amount of light. But then afterwards, it can basically make a recording off of how much of that light is blocked, and the more light that is blocked, the greater the turbidity, and because of that, the greater the growth. So that is kind of the best way in order to score them. It's going to give you the best data points that we have. However, again, for our class, using our just basic scoring this way is sufficient as well. But you should be aware again that there are 3 basic ways that we can score it. Either kind of the yes-no basis, did it grow, yes or no, can we use our turbidity scoring, or can we actually use a spectrophotometer in order to get these nice, accurate, true data points that we have for a specific setup? But really, I think other than that, I don't have anything else that I need to introduce for our temperature and pH experiments that we're going to be doing in class.